Okay, so we'll get started, and then we have a couple of demos and we have a couple of Slidos for all of you. Like I said, we know it's the last session of the day. We know it's been a really long day, so we would just love to have you all be engaged, and then maybe we could get out of here a little early too. But so we are here to talk to you about this session, You Are What You Eat, resources to address the need for metadata-rich, high-quality biological data to feed AI applications. So we're going to be walking you through this session with these different features. So every part of this will feature open discussion and some Slidos. We won't do any breakout groups or anything, but we're just hoping that the whole time we'll have some live engagement. So we'll give you a little bit of an intro and then some different reporting standards. We'll talk to you about the National Microbiome Data Collaborative that Montana, Samantha, and I are a part of. And then we'll talk about some of the importance of metadata and metadata standards that you've heard about throughout the week as well. Then we'll do some demos and then have a final Q&A here. So again, this session will be as interactive as you all will allow it to be. So please interrupt me, ask questions. If you have things, if you disagree with anything that we're saying, we're really open to having whatever discussions you all want to have, and then the direction that these discussions take can be up to all of you. And I will reiterate that if you ask a question or provide your input, we will provide you with a piece of candy. So just to influence all of you to do that. And then we encourage you to take out your phone or laptop for the demo or hands-on portions. Montana will walk you through some of those, and we would encourage you for the Slido during Montana's session to join on your computer because we have some things on your phone, so it might be easier to have the Slido on your computer. And then again, feel free to interrupt us, won't take it personally, and then feel free to say whatever you want. But I'm Julia Kelleher. I'm at the Los Alamos National Lab in beautiful New Mexico. I'm also affiliated with Michigan State University, and I'm the Outreach and Engagement Team Lead for the National Microbiome Data Collaborative. And
I'm Montana Smith. I'm at Pacific Northwest National Lab, and I'm at the Environmental Molecular Sciences Laboratory, which is the DOE user facility. I'm the submission portal product owner for the NMDC, so all things metadata, data standards, data collection. And while PNNL is in Richland, Washington, I actually reside here in beautiful Seattle.
And then shout out to Samantha Miller, who's helping us online, and she's also a wonderful NMDC team member as well. So thanks, Samantha. Any questions about who we are or anything so far, or clear so far? Great. Okay, so first will be the Slido that we'll use throughout the session. So again, our session is You Are What You Eat, talking about data, but also the candy that we're eating during this session. So this is where we just want to have some discussions with all of you, have some questions up there. So you can either use the hashtag or use the QR code here. Does anyone need any more time? Looks good. Perfect. Great, great, great. Okay, so we've all been to, or if you've gone to these sessions, we've heard a lot about this phrase of AI-ready data, and it seems to mean very different things to different people, as we've heard. So I have tried to, in the next slides, spoiler alert, I have some quotes that we've heard throughout the week of how people define AI-ready data, but we're curious as to if you were to think about AI-ready data, what's the first phrase or words or qualities that come to your mind when you think about AI-ready data for your environment, your data, and your applications? So all of the metadata, love that. Montana, I'm sure, loves that. Data are harmonized, standardized, machine actionable, annotated, fair, rich metadata, clean, readable, formatted, organized, minimally heterogeneous, well-documented, love that as well. So we're seeing some commonalities and then some that are a little bit different in the way people interpret this. Lots of clean, minimally, yep. So I think these are all great. You can feel free to keep adding things here, but I think we can go to the next slide here. So the commonalities, so these are kind of the words that we've heard the most, at least from our space and then from some of the sessions earlier this week. We covered the high quality, the completeness, free of errors, the consistency, and standardization, metadata rich, accessible, fair also came up, structured properly for the specific application, proper file types, verifiable results, proper training data sets we'll talk about a little bit too, and then sufficient data and metadata for valuable insights, And then we've also seen other quotes like this of there are these AI ready checklists that have come up. That was a really nice session. So people define it as things that perform well against these checklists. We've also heard all data is AI ready data, or no one knows is my favorite. And then, obviously depends on what you want to do with it in the AI applications. But I guess how do people feel about this all data is AI ready data? Are people in favor of that? Negatively? Yeah.
Oh, yeah, okay. I mean, I'm not saying I agree, but I think we have to make that, like, assume that that premise is true. Like, if your data is online, and AI is gonna come for it, whether it's ready or not.
AI is going to come for it, yeah. Other thoughts of all data as AI ready data? Yeah.
Just the first thing that comes.
Oh, sorry, here comes the mic.
So going along with this, you are what you eat. The first thing that kind of comes to mind when you say all data is AI ready, it makes me kind of think of the phrase all mushrooms are edible at least once.
Yeah, other thoughts? So we had a couple of discussions throughout the week about this too. So this is something that we've gotten as feedback of, you all are structuring your data too much. Just throw it in, AI will figure it out. And our response is, what we'll be talking about in this session of you can do that. The quality of what you get out will reflect the quality of what you're putting in, which inspired us to put in this session proposal to kind of advocate for these standards and this metadata richness, because we do believe that that will create the highest quality AI insights. But we do know that there are going to continue to be these critics, so to speak, of just saying all data is AI ready data, it will figure it out for us, and everything can kind of be funneled into the machine. So I think keeping that in mind of these different perspectives as we go through this session too, we can play a little bit of, I guess, devil's advocate for things, but we will be focusing on these two specifically throughout the session of having this high quality data with rich metadata that's complete and standardized. So we'll be talking about the benefits of that, and then some resources that can help you with that, and then some opportunities for giving us feedback on things we've developed, and then how we can work across scales, across data types, and everything like that. So I personally am not the believer that all data should be AI ready data, just putting that out there in case that wasn't clear from what we were saying. And then also, this has come up a few times throughout the week too, of people thinking just about the end result of putting things into a fully fledged AI machine and getting those outputs, but then what are you using for your training data sets? Are those high quality? Are those metadata rich? And when you're retraining your models, is that something that is being considered in terms of the quality as much as that end result? So understanding the quality of the data that went in to train these models, and then thinking about the quality that you're getting out of those. So that's something that we added later, hearing other sessions too of the importance, obviously, of these training data sets, and then how we're thinking about preparing for this next level of AI applications that are on a global scale or across different data types, and then also ones specific to our own data types. Okay, so then as I kind of alluded to, we're going to highlight some tools and resources that can help you collect, analyze, and report on metadata-rich data, but then also just provide a basis for discussion. This is not a Slido question, sorry.
Which means it stopped recording. Oh, stop recording. Okay, hang on. I don't know who kicked me out.
This says on here, Zoom Workplace signed out. You've been signed out because you're currently signed in on another device.
Yeah, that's what it just did to me.
Okay, so now it did it to me here too.
Okay, hang on. I'm gonna, God, what?
It still says it's recording?
Oh, well, okay, but it kicked you out?
I think, but this is you. Yeah, so you, I think, are signed in on both, maybe?
Because I can't click start. What happened?
Yeah.
Yeah. Yeah, this is still online.
Everyone else is still online, can see you and can see slides.
Yeah.
Because I'm logged in and I can see everything. Well, okay, if it's still recording and everybody's still on, I will.
Oh, yeah. Still says pause /
Yeah.
stop recording.
Are you the host? Someone else commented that they can see and hear.
Oh,
Okay.
great, okay, awesome.
It says ESIB fellow support. That's maybe, is that supposed to be? Okay, well, you're
It says you're in there
still
somewhere.
here.
Maybe there's a third
So
version
that's
of
funny.
you.
Oh, what? Okay. We're good. Oh, thank you. We are waiting for that. Would you mind if I ask you a question?
Oh, of course, yeah.
I think we are okay. I don't know why I did that. That was weird.
I'm just wondering if you go back to the slide, the previous one, if you want to distinguish the AI-ready data and the training data, because it's very highly likely we will not be able to make all data high quality and metadata-rich, but obviously training data is very important and critical.
Yeah, completely agree. And yeah, we added this as kind of a conversation starter for this as well. So I'm glad you brought that up. And then in a session earlier, they kind of talked about those benchmarking data sets too, to make sure that for each retraining session, you're able to re-verify those results from these ground truths. So having those be your highest quality, most verifiable, most metadata rich, I think is a really important point, that your training data set should be kind of your gold standard for things. And then starting to feed in some of these that are more realistic for what you'd be seeing, and then seeing how it works with some of those. Yeah, someone brought up in the session earlier of feeding in something that is really metadata rich, of even strings, and saying rainfall was this amount in Paris, and then seeing that it's saying, of course it means Paris, France, but maybe it meant Paris, Texas, and then kind of working these little things and correcting things as you go, but kind of starting with, yeah, most metadata rich for the training data sets, I think is a really good point. Did you provide candy? Okay, great, great. Most important. Any other questions, just before we kind of get into some other, yeah.
Maybe it's too big, but I'm just struggling with this a little bit, and always am with the AI ready, is and I think, yeah, you have the for the application, like that's the one I'm like, yes, for the,
Yeah.
as in a particular application, and when you take that out, then that's like saying even human ready. Is it
Right.
human ready? Well, who's the human and what do they want it
Yeah.
for? Maybe, maybe not. And so I guess in the context of, well, the rest of the session, should we be thinking about in a context of an application, or is it some minimal AI readiness that's application free, and then maybe there's something on top of that, or?
Yeah, so that's the perfect segue to our next set of slides of this session's focusing on biological data, but then specifically microbiome data. So how we kind of approach AI readiness and what we kind of think of. So we'll be giving you some use cases of what we're doing to kind of create this metadata-rich data for microbiome data sets, and then where some of those pitfalls have been from our own experiences, because we can't speak to any of the other data types that a lot of you all work with. So that's kind of where we're coming at this from. But then I think having this was more of a, these are all of the different things that we've heard, and I don't necessarily know that one thing is the right thing on this list, but I think having it be application specific certainly seems to be the most all-encompassing definition that is kind of, you could put poor quality data in if you're happy with poor quality results kind of coming out. So I think that's a really good point.
I think I'll just add to kind of like get a little more specific on your question about, application. As like within NMDC, we're just starting to kind of explore into this AI application side too. So I think part of what we want to hear is what would be the applications that would be most applicable for this community.
Okay, so your perfect segue takes us to this slide, which is we're going to be talking a little bit about biological data. So again, this is just because Montana and I both work in this space. We work with biological data, and this is also a plug for this EMSL, the EMSL, Environmental Molecular Sciences Laboratory. They have a session tomorrow that it would be great if you all could attend. And so they kind of work in this space that they have circled here, and you'll see this same figure, I think, tomorrow too. And so they work with a lot of these different data types. We are more on this bottom corner side. But we wanted to present this to you because we're talking about microbial data for a lot of this, and we know that a lot of you don't work directly with microbial data, but you can kind of see from this how quickly things can compound from genes to proteins to organisms, and then to entire environments and ecosystems and to the global scale. So we try to do a lot on the continental scale biology. We try to scale up from the microbes as much as possible. So we're really interested in hearing a lot from you in this session of maybe if you don't work at all with microbes or think of things on the genetic scale, how can we then work with some of your models or data types to integrate across these scales and across these systems? And we can talk through some examples. Other ones that we've talked to folks about earlier this week is satellite imaging being used to identify harmful algal blooms, and that's a microbial phenomenon, and how microbes play a massive role in nutrient cycling that directly feed into things like climate models. So we encourage you in the next couple of slides too to think about whatever data type you're working with. Maybe microbes would be really important to consider and what they're doing, since we can have functional data about what they're actively doing in their environments and the products they're producing, and thinking about how we can find those commonalities between the data sets, even if it's just latitude and longitude for trying to link up different data types. So if it's also not clear, I love microbiomes and microbes. So we study bacteria, fungi, viruses, which some people consider microbes, some people don't, and archaea and other just small little organisms. So my favorite part of hosting workshops is convincing people that microbiomes are super important. So how many people have heard of microbiomes? Gut microbiome? What about other microbiomes? Really just the gut? Where else do people find microbiomes? Where do you kind of think about them? Yeah.
It could be on the skin or fur or whatever of an animal.
Yep. Yeah, really good ones. Where else do people just think about them when they think about microbiomes?
Soil.
Soil. Montana's favorite.
I'm gonna guess maybe bromeliads or pitcher plants. They probably have microbiomes
Yeah. Yeah.
in them.
Where else for microbiomes can you think of?
Or care about.
Yeah, where would you care about a microbiome? Where do you think it would affect you too?
In a hospital or in an air handling system?
Yeah, yeah. Tons of microbes that get in there. Ralstonia piquetii can get into tubing, gets right into people. Yeah, gnarly one. But yeah, basically, if I told you all to guess, you would probably guess an environment that has a microbiome. So for plants, it's the roots, it's the leaves, it's the shoots, it's the soil surrounding the roots. Each of those has a different name for their type of microbiome. The air has a microbiome. If you swab some surfaces, I've unfortunately swabbed phones and keyboards and found quite a diversity of microbes there. Disgusting, I know. And it's really, yeah, everywhere that you can think of. So if you're working with oceanographic data, the marine microbiome space is really interesting. Deep oceans have microbes in there as well. So really thinking about each of your data types, I bet we could find a microbiome that overlaps with you in some way, and just thinking about how we interact with millions of different microbes, billions of different microbes every day, and kind of everything that we do, built environments. So yeah, everything in buildings, subways have been swabbed quite a bit too, also astonishing. But yeah, just kind of taking, if you aren't someone in the microbial space, maybe thinking about how you could be. And again, these directly impact soil biogeochemistry, human health, animal health, plant health, and then even these macro scale systems of what's going on a global scale. And something that I do want to highlight is there's also an, again, astonishing amount that is not known about microbiomes. So these microbes are doing a lot, and we know that they're doing a lot, but we oftentimes don't know what they're doing on a second-by-second basis, or why they're doing what they're doing, and how they're responding to their environments. So oftentimes with microbiome data, you get this massive black box of there are these little things that you know and these microbes that you know, and then there are oftentimes dozens of microbes that have never been found before, or proteins that you can't identify because they're not in a database. So there's a lot of this information that is really difficult to parse through manually and to try to figure out what's going on. So that's why we feel like there's this massive potential for AI applications to start to parse through this information and then start to make some of these connections connections with what these microbes are doing in their environments. And then metadata, you all know this, but metadata is extremely critical for everything, but especially for microbiomes. Even a tiny shift in pH or the addition of one microbe or an extra bit of sunlight can completely change the entire microbial community. So having really strict metadata standards and how you're reporting that is really critical for these really small scale applications, like looking at second by second proteomics or figuring out one soil sample that's 6 inches from another soil sample of why that might be completely different, and having that metadata is really critical, which Montana will talk about. Okay, so we've kind of talked a little bit about this, but do you work with microbes, microbiomes, or microbial data? There's no wrong answer too, you don't have to say yes. Indirectly? Can someone give more context to what they mean by indirectly?
I'm an ecologist and data manager and work, so I don't do any kind of like molecular or microbial ecology, but I work with people who do, and just I help them publish their data that's kind of environmental context for the sequencing data and stuff that they put in, NCBI or something like that. And I will say, I'm shocked by how little metadata you can sometimes get back from a platform like NCBI. So I hope we can talk about that a little bit.
Yes, we certainly will. That's my favorite thing to talk about. Anyone else want to elaborate on indirectly? Did you raise your hand? Sorry, didn't mean to point at you. Okay.
He said what I was saying again more, so.
Okay.
I'm in a similar role.
Microbial ecology?
No, just helping.
Data manager.
Publish their data.
Yeah, great, perfect. Others, indirect, or people who are interested? Did this session make you more interested, less interested? Don't want to know what's on your phone? Okay, so some people working with microbial data, maybe some people that might in the future. And then we do just want to know, what data types are you working with, just so that we can get a sense of who's in the room, what data types you're working with, what environments, what data types. I think this should make a word cloud. Montana's our Slido expert. She's helping out. Omics, soil. I suspect that came from over here, maybe. Whole organism counts, cool, yeah. Mass spec, ocean. Hyperspectral, cool, remote sensing. Plant ecology, lots of ocean. Generic data arrays. Rock samples have a lot of microbes. There are fungi that can digest the rocks slowly. They eat all the minerals from that, super cool. Coral, the coral microbiome is huge. Hyperspectral data is also now being used for microbial, so you can actually scan into the soil and you can see the presence of certain fungi, especially ones that can make different pigments. So they're being used as a readout, and then also ones that can add different fluorescent tags into plants. So those are pretty cool. So just thinking about adding microbes, clinical, yep, huge deal with infection, plant community, meteorological, yeah. Soil, is that a data type or is that a soil typo? Okay, typo, okay. But I love the idea of a soil microbiome. Yeah. So huge diversity here. Also just want to recognize that that's really cool to be here in this conference. I usually go to microbial conferences, so this is just a really awesome way to meet people working with different data types and in different environments. So yeah, really cool diversity here. Productivity, that's cool. Yeah, lots of ocean. Another plug for ocean microbiomes. They're really cool. Okay, so then now that we kind of have a sense of who's in the room, and we've talked about this a little bit, but maybe someone who hasn't spoken up yet, how can microbial data maybe relate to your data type in a way that we haven't talked about? Don't forget Candy is on the line. Did we cover everything of maybe all the different applications we could think of? I think I gave as many examples as I could come up with in my head for different microbiomes. But hopefully maybe people are thinking a little bit more about microbes in their data sets or their environments. Yeah? Okay. That's all we wanted to accomplish from this section, so that's great. So then coming back to AI for our use case of microbiome data. So these are kind of some examples of how microbiome data has benefited from AI usage, and then how, just some examples of what we've heard from the field of how they're planning to use this, or how they think AI is going to be a part of microbiome research moving forward. So some of these large-scale meta-analyses, so NCBI has millions, billions of data types, or data sets rather, for microbiome data and metagenomic data and metatranscriptomic data. And there's a lot to parse through. So these really large scale meta-analyses, AI is helping with what questions can we ask and answer? How can we visualize this data? What can we do to do cross-study comparisons? And then looking at these large scale modeling efforts. So something that I think this room would be interested in is some of this last piece too of helping to connect data at different scales and different data types. So looking across different data types, AI can help with these translations of using different terms, working with different data types and environments, and how do we connect one with the other when the scientists may not speak the same language, literally or figuratively, and then having AI kind of help make those connections and figuring out how to integrate those different data types. And then there's also really cool things like the tool that can take in genetic information and then use AI to help predict what the protein folding will look like, and things like that on a really small scale that have been really impactful for how we understand things at the small scale so that we can build up to some of these larger scale models. Okay, so then you're all here because you are what you eat for this session. So we've talked a little bit about this already, but it really is this garbage in, garbage out. So when I first learned how to do sequencing, this was something that was talked about a lot in terms of your DNA quality and your sample quality. So we just wanted to kind of say how this continues to compound with these different steps. So for our use case, for our workflows, if you do a poor job of collecting your sample in the field, so maybe you're doing an ocean experiment, you contaminate it on the way, or you spill half of the water, or things like that, you can lose quality there. Your DNA extractions, your library prep, your bioinformatics analyses, you kind of keep having these compounding stages of garbage in, garbage out. So just the importance of QC at each step, which I know has been talked about in other sessions as well, that continues to compound as you get to these final AI steps as well. So having proper QC at every step to know that what you're putting in is something that is accurate and that it's high quality, which we also are defining as also kind of an arbitrary definition. So we also wanted to open up to all of you of how you kind of define high quality and low quality in your data sets if you were to just summarize it yourself. So we usually have, actual QC metrics for certain aspects of what we do. But then generally we would say that for us, low quality data means sequencing errors, lack of standardization for the data or the metadata, incorrect metadata. Our favorite SRA example of lack of metadata. I was doing a meta-analysis recently with about 80,000 SRAs, and pulling in all of the metadata, it looked like an error that everything had NAs across the board for the metadata for even things like geographic location or host.
I was just going to ask what SRAs are.
Oh, yeah, great question. So NCBI SRA, so NCBI is the National Center for Biotechnology Information hosted by the NIH. The SRA is the Sequence Read Archive. So basically, it's a big archive of sequencing data. So a microbiome can go through and be sequenced, or an organism, so even like a human, if it's sequenced, you get the genome information, so all the A's, T's, C's, and G's. All of that genome information could get dumped into this SRA. It's a data repository that's kind of the gold standard for where sequencing data goes. That's accepted by all journals that kind of publish sequencing or omics-based publications. But so that's, they're a little bit notorious for not having great metadata or not rigorous metadata requirements, because they also want to be open for if someone doesn't have a lot of the metadata, but that leads to a lot of these issues of blanks across the board. And if things aren't enforced, I'm maybe a little bit guilty of this sometimes, if it's not required, I don't always put that information in, but I have improved that as I've, aged. But I think it's also something that is important to consider of we don't want to be too onerous on the user of having too many requirements. But then if we do not have those, we see these examples where now there's millions of data sets that are not meaningfully reusable because they don't have any of this metadata that would make it reusable. And then this one came up in a couple of sessions of just making sure there's no malware in things too before you feed it, or just bad things in your data. That's also something that's interesting to consider. Make sure that's not in there. And then just to see if there are commonalities between what you would consider low quality data for your own work, just seeing what we get there. And anyone can speak up too if they want any candy. And I'm also interested to know if your definition of low quality data is a strict definition, or if it can vary in different instances, or if you have a sliding scale of low to high. Minimal metadata, poor metadata, rounded for no reason, nice. Not enough replicates, also a really good one. Low accuracy, low precision. Blurry images, yeah. Yeah, and one thing that Montana has been grappling with is it worse to have incomplete metadata or inaccurate or bad metadata too? So that's also maybe a thought question for all of you. Low resolution.
I'll add to that inaccurate or lack of metadata question, which is something that we're thinking about for NMDC is how can AI help us complete our metadata, right? So if I give you a publication or an abstract, can it, extract out the metadata? And if it does do that and it pulls the wrong thing, what's worse, right? Let's say that it says that I have a temperature treatment and it's a below ground temperature treatment, but it registers it as an air temperature treatment. Is that better or worse? And how well can we trust AI? Oh, Kelly, you wanna add anything?
At least in that example, I would say worse, because from my standpoint, so my background is statistics, so I would be trying to use that metadata for an analysis to make comparisons, possibly based on those treatment groups by temperature.
Lack of data dictionary, that's a good one too. Another hand. Inconsistent IDs.
Yeah, I would agree with that. Having no metadata is better, or lower metadata is better than having incomplete metadata. We're struggling with that at NOAA a little bit right now. We have a rubric to encourage people to fill out their metadata, but it is a completeness rubric. So if you put in bogus information, you get a high score. So we're really trying to fix that. And to make it really clear that we want good quality, even if it's less. But kind of like you said, if AI needs that metadata to be complete and accurate, then providing nothing or a couple of fields is not going to help. So yeah, it's a hard thing to solve.
Yeah, and Montana's struggling with this too. We've also been tasked with creating a metadata rating system for our data too. I think it's maybe 1 to 5 stars or gold, silver, bronze, something like that, and trying to figure out what to penalize people for, because we also understand that for Indigenous data sovereignty, you can't always publish the exact coordinates for latitude and longitude for data, and being respectful of that while also kind of mandating that field as much as possible. We're kind of trying to weigh how to weigh completeness with quality, with things that are justifiably not included and things like that. So trying to make it less of a manual process, but then also having some type of standardized rating system so that people can know this is really metadata rich. This would be perfect to train your data sets or train your AI rather with these data sets versus something that may be a little bit harder to train an AI application with or an agent, and then maybe have a little bit less confidence in. And then leading into this next one, so this is also an open discussion. So we've talked a lot about this low quality data. I've definitely been guilty of generating low quality data. Things go wrong, things just happen. What do you all do with your low quality data? Do you just throw it out? Do you cry about it? Yeah, and that definitely happens. We've been there. You try again, but what happens to that data? Do you continue to store it? Do you throw it out, never do anything with it? Do you publish it alongside your other results? Do you still make it publicly available? What do people tend to do with their lower quality data? Yeah.
I'm not in this field, but I always rely on, the archiving system. Is like, is it a working document or is it something that we need to save? So it's just sort of like, what's your protocol? What's the regular protocol for that? And so I sort of try to rely on those. I wish I could think of the terms right now. Yes, based on the archiving system. And what do you deem? Are you deeming that data set you collected, are you deeming that a working product or are you deeming that a final product? And so how do you treat those two differently? And so that's what I try to rely on. So if you can set a standard that way, and try to rely on that, I guess is what we're here talking about.
Yeah.
I think I'm a little confused about what is low quality data, because when we normally think about low quality data, we tend to think it's scientific quality, but now we're also thinking about metadata documentations that's more associated with the data product. So if it's scientific quality of the data, we trying to validate the value, and that's kind of like the validation part. And that would tag the data points or data values that in question. So people who actually use it and know this particular one, you want to pay attention. But if it's a metadata or description part, the repositories or data curators could help, and then data producers would also follow the guidance that provided to them, and because they're the ones have to provide information that's accurate, right? So in terms of quality accuracy, you could be data accuracy, you could be metadata value accuracy. And for example, if it's location are half degree off, it's very hard to capture by automated tool, but that where the domain expertise would become very important.
Yeah, well said, and I should have had a better transition from the last Slido, but having low quality data being however you define your own low quality data for these different applications. And I think, yeah, what you said makes a lot of sense, and how you kind of define that and the different, scientific accuracy or scientific quality of things. But yeah, sorry, didn't mean to interrupt.
No problem. I guess maybe it's also a continuation of that, the definition of low quality data. So I'm an oceanographer by training, so I work mostly with ocean circulation models, and I've been charged sometimes of producing the data. And one of examples of quote unquote low quality data that is still publishable happened within our group is that we were interested in this particular area of the ocean, and then the physics of this particular year. And so you take large scale mean fields to force a model to produce data. And you're hoping that the mean fields will replicate the mean fields from observations, and you look at the turbulence aspects of things, or lower scale resolution. And at the end, we do this simulation, and then the fields were wrong. The ocean was significantly saltier than it should be for this particular time of the year, for this particular region. But you can still do science with that. And there was a lesson learned. We didn't struggle to do or to think about what to do with this low quality data because it really wasn't, the mean field was wrong, but it wasn't unphysical. It just didn't exactly match what we were expecting for the mean fields. And so, well, we can at least this kind of scenario explain what's going on regionally. And so it became a publication and we made the data available. And so it's important to just, if you make a mistake, just kind of like disclose it in this type of metadata and make it publishable and share that as well. That's a lesson learned.
Yeah, thank you for sharing that lesson learned. I think that's, yeah, an important note of how low quality data can become high quality data and still usable in some senses. And yeah, great point. So I know I'm running short on time too, so I'm going to speed through this, but this is then talking about your high quality data. So how do you then make that available? So we're going to be talking a little bit about reporting standards and some reporting guidelines that our group has developed, but then thinking about what guidelines or standards are used when you're publishing your data into repositories or in scientific publications. Do you all use reporting guidelines or standards, especially for your publications? Yes, no, yes.
Raise your hand if you do.
Yeah, who uses reporting standards or guidelines for when you're writing a publication? What about when putting into a repository? I kind of just, follow the repository guidelines or the other standards. Yeah, okay. So yeah, we're just gonna be talking about reporting guidelines, and when we talk about this, in this context, we mean in a scientific publication. So we've come up with a set of standards for microbiome reporting that we'll talk to you a little bit about, but I would recommend thinking about for your own data types, what reporting guidelines exist out there for your specific data types that can also cover the metadata that you should be reporting in your manuscripts to make sure it's fair and reusable, make sure you're citing your data properly that you've reused. So basically, there's this checklist that existed for human microbiome research that was published, and then we convened a workshop last year with 50 microbiome stakeholders, so researchers, funding agencies, data repository representatives, and missing one other key group, researchers, funders, data repositories, journals, publishers. So we got together with 50 of us in a room to kind of talk about the state of microbiome reporting in these manuscripts, to talk about what was needed to maybe transition those STORMS guidelines into something that we were calling the STREAMS guidelines. So we have a workshop report that's available if you wanted to hear about this. So I had a poster on it yesterday that, I probably should have taken my poster down, but it's still up right now. But unless they took it down and threw it out. I got interest yesterday and chatted with some of you about just the process of creating these standards too, and got requests to maybe just talk through how we kind of came to this consensus and what the workshop was about, and creating a set of reporting guidelines. But we came up with a first draft, and then we circulated that first draft. We got over 700 comments on that first draft. I responded to all of those in a response to reviewer style document, one at a time. Would not recommend, but would recommend for the people that got the feedback. They appreciated it. I just did not appreciate myself typing at 4 a.m. trying to respond to all of these. Then circulated the second draft, got 400 more comments, so 1,100 total comments. And then we had over 250 researchers provide us with feedback across 30 countries. So we're really proud of this response rate. And then we really felt like we could build a community consensus set of reporting guidelines based off of the STORMS checklist that already had quite a bit of community buy-in across journals and publishers and funders, sorry. So we really tried to build off of that and create this community-based standard. So the STREAMS reporting guidelines are more for environmental microbiome reporting. So we have 67 reporting items that are organized like a scientific manuscript, so abstract, introduction, methods, discussion, conclusions, and then other information, so supplementary files. So we really tried to mimic the architecture of a scientific manuscript, because we also heard that that's the most beneficial for students, especially when they're writing their first manuscript into this area, and they're really trying to navigate what do I need to include for my publication. It's in review at Nature Micro. We submitted our response to reviewers about last week, so hopefully hopefully that goes through. And then the workshop report I'd posted earlier. This is our little website if you want to go. This is a teeny tiny font, but so basically this is a little snapshot of what the guidelines look like. So we have an item number, the item name, the kind of core recommendation, the source that it came from. So if it's an existing metadata standard or an existing guideline, or even things like SRA for biosample import, that's something that students struggle with too. So we wanted to provide interoperability across existing standards for repositories too. And then columns here for saying, is this in the manuscript? Yes, no, or maybe. And then what is kind of, or where it is in the manuscript. So we also wanted this to be a resource for people reviewing manuscripts, because it can help sometimes to have a checklist to go through and say, wow, they didn't report on their permit information, or they're missing the DOIs of the data sets that they cited, or they completely neglected to say how many people they sampled or environments they sampled, things like that. So trying to help for students, researchers, and reviewers for manuscripts. If you want to get involved, if you do any microbiome stuff or want to be involved, we're also building an LLM to parse out manuscript information for both the storms and streams guidelines, so that you don't have to do it yourself, but it would be something that can kind of pull out that information for you, and then you can quickly take a look at that. But if you would like to contribute to that publication, let me know. We're having humans do examples that we then compare to the large language model that's parsing this out, and then look at the similarities and differences in which items are performing well, and then if there are differences and things that can be improved. Okay, I really have to speed up here. Yeah, anyone quick challenges with things that you've seen reporting in manuscripts for your data types or your environments?
Thank you. I actually have a question. Sorry, to go back a little bit. So I don't publish pretty much any manuscripts or scientific data. I help data managers get to that point. But so each journal has different sets of requirements, right? So is it clearly indicated in this, for like Web of Science, like which journal this complies with? Or is it like an amalgamation of all of them? And it's just, if you fill all this out, you'll be good to go on any publication?
Yeah, that's a great question. So we say that the organization and kind of the structure is totally dependent on the journal. The key information, we tried to keep it broad enough, and we do have some kind of parentheses in there of like, if the journal requests this, or if this is, if the journal will allow things like reporting on the manufacturer of kits. That is kind of journal specific sometimes for things. So in the actual, we have a tutorial video and a user guide. We kind of explain how this could be applied across different journals. And then we always kind of say defer to the journal specific requirements for the final version of things, but this is supposed to be kind of a catch-all. And then we do have a lot in the supplementary or other information that we think should be captured somewhere, like AI usage, but we also say that that's extremely journal specific right now of how you're required to report what you used AI for in your manuscripts. Yeah. Okay. I'm going to skip over that really quickly so I can speed talk. We're also with the National Microbiome Data Collaborative, as we said, trying to connect data, people, and ideas to advance microbiome innovation and discovery. And so we want to create infrastructure for microbiome researchers and also connect with people across all different environmental sectors to make microbiome data available to everyone. So all of the data sets we have free, open, available. If you want to reuse some microbiome data sets, we have everything in our data portal that we will show you here. Everything is publicly available. And we run everything through standardized bioinformatics workflows. So everything was processed in the exact same way. So you don't have to reprocess data. You can download all this multiomics microbiome data, and we welcome you to reuse it. So we also thought that, especially with different data types, you might be interested in kind of exploring where you might have samples collected from, and might be interested in what microbes are there. And then we have all of our extensive metadata fields that you can search by too, like pH, what else, Montana? Even like microbial functions.
Environment
type.
Depth within soil, yeah.
Collection date, yeah. And then the different omics types that are available. So yeah. Online question.
I can share. I guess everyone online can read this though. Stace was asking, have the items in the streams guidelines been matched or crosswalked to any metadata standards like MixS or Darwin Core DNA derived data?
Yeah, great question. So in that item source column, Montana actually helped me to pull out the exact what's it called, Montana, item number or the MixS, the term, that the full term
ID.
and the term ID is actually in the item source column. So it's directly matched with MixS and the Genomic Standards Consortium, and then also directly matched up with the NCBI biosample and SRA import information from their spreadsheets. So we tried to make it as interoperable as humanly possible.
And I'll just add, since Darwin Core was mentioned, that there is an effort to map MixS and Darwin Core, and that's an active program.
We also have extensive community building programs. If you want to be a champion or an ambassador, I can tell you what that means later, so I don't take up more of Montana's time. But we have a lot of engagement programs. Samantha's leading our user research effort too, so we take user feedback into consideration for everything that we do. And then we have really cool programs where we engage the community. So if you're passionate about fair, open microbiome data, or just data related to microbes, let me know or chat with me and we can connect you with some of our engagement programs. These are just some good resources if you just want to learn more about the NMDC. Montana's going to talk to you some more about some specifics, but these are some links. And then to our website and then our social media, where thankfully it's not me designing everything for social media because I don't know what I'm doing on social media, but we have a wonderful social media person who's making all these cool graphics and things. So feel free to follow us there as well. And then Montana's going to be walking through metadata and some demos.
Okay, yeah, so I'm going to walk through just a bit about metadata, what we do on the scope of metadata, and we also will do some hands-on workshop-y stuff with some of our submission tools that you'll use your phone and your computer for, if you are so inclined. Okay, so this first, just tell me, you do define metadata. Within your area of research or what you care about, how would you define metadata? Data about data, yes. Data about data. One scientist's data is another scientist's metadata, yes. Especially in our area with microbes, right? pH, carbon content of your soil, all of that is data, but we often treat it as metadata when thinking about the microbial community. The context, yep. Details about how samples were generated, analyzed, and how the data was processed. Yeah, that's a really big one. We are thinking about a lot within the NMDC is the importance of, especially when we think about mass spec data. I think I saw a small word on there for the word cloud about mass spec data, where how you extract your samples, how you set up your instrument, what instrument runs, held what samples is really important to understand, to know how interoperable and comparable your data really is. Oh, there's another question online?
Yeah, the question online is, to submit to NMDC, do you have to be funded by DOE?
You do not have to be funded by DOE. So NMDC is open to everybody. Everybody can put their data and metadata into NMDC. A little bit of a caveat, which is that, depending on how much help you might need, there may or may not be more levels of support for DOE-funded programs that are focused within specific DOE calls, right? But we have a ton of NEON data in the data portal right now, and more coming, and that is not DOE-funded. So no, you do not have to be DOE. All right, I'm gonna move on. So metadata for NMDC. Yeah, contextual data about your data. So vital for the data's publication, depositing your data, preserving your data, making sure that it can be discovered, accessed, and reused. So it's everything that's really important for the data, yeah.
I was just going to ask a question. So when you say you can put your data in NMDC, you're talking about raw sequencing data, essentially?
No, so we're not a repository. Okay. Sorry, yes, I should make that clear. So we are not a repository. We are more of a discovery tool. So if your data is available on, like, again, NCBI or wherever, we will pull that data, process it through our standardized workflows, and then provide that data in so that you can download NMDC data and compare across all programs, or that in all studies and all data within NMDC. So you know with confidence that everything's been processed the same way, but we're not a repository. And then specifically sample metadata. So this is the when, where, what, how your sample is, how you collected it, what are the treatments that were applied, what environments did you collect that sample from, again, about microbiome data. So this is what we're talking about when we talk about sample metadata, which is important to define because if you've ever, thought about your specific area of research, depending on who I'm talking to, a sample for them is the extracted DNA. Whereas for us here at NMDC, the sample is the soil from which that DNA came from. And the DNA is the more of a process sample that's gone through some sort of process to prepare it for an instrument. And metadata is important everywhere. There are lots of different types of metadata: sample metadata, environment metadata, biomolecule metadata, again, that processing metadata that I talked about. And it is important everywhere. The submission tools that I will walk through are focused primarily around, again, samples and environments. And within your data lifecycle, you have to capture your metadata at every step, and it's important to think about it early and often and plan for it. So if you miss a step, then something could be misconstrued. And this is really important within that AI application area. If you miss a step within your metadata capture, and we are trying to get a machine to use that information, it may not actually understand or know exactly what it is. And we've all experienced hallucinations and AI lying to us, and so we want to make sure that we can trust what we're being told. Okay, so metadata standards. NMDC has implemented standards for capturing metadata, and we did this because when we first launched our program, we kind of pooled as Julia said, we're super community driven, so we pooled the community to ask what are some challenges within microbiome data reuse, and what is your biggest challenge for sharing microbiome data? And it all boiled down to the metadata, which the text is also kind of teeny tiny, but can't totally read it. The first bar on one side is missing or incorrect metadata, and the top bar on the other side is difficult to format metadata and data submission. So I can't use your data because the metadata sucks. I can't give you metadata because it's hard to follow these guidelines and follow these standards and understand the expectations. So we're kind of at this bit of a chicken and egg situation where, like, which one can we solve first? And there's a lot of consequences to not standardizing your metadata where you, again, miss critical information. Data can't be deposited to certain repositories because they have certain requirements. You end up with a bunch of NAs. You can't reproduce that data. So all kind of similar problems that we've been talking about throughout this session. So another Slido question: what kind of standards or ontologies, or what standards do you use within your research domain? So part of the reason why we wanted to host this session was so that we could see the bridge between NMDC microbiomes and just Earth system in general and more broadly. And so we focus on a few standards within our system, but we make it a point to map to and link to existing standards outside of what we are aware of. So if you have a certain standard or ontology that you use more commonly, then we want to make sure that we have that connection. Darwin Core, big one. LinkML, I'm not going to talk about it much here, but our metadata, our NMDC schema and our metadata schema is actually LinkML. It is a LinkML schema, and we work closely with Chris Mungle for all that work. EML, ecological metadata language? I don't know that one. ISO, CF, OGC. What's OGC stand for? Open Geospatial Consortium, all right. Nice. CF. Awesome. Give it another couple seconds. Oh, more EML.
Does anyone not use any standards or ontologies in their work?
If it's a no, you can write no.
Yeah.
That's
You can
okay.
rat yourself out.
All right, I'm gonna move to the next one. So at NMDC, The metadata standards that we use are the minimal information about any X sequence from the Genomic Standards Consortium. We also use the Genome Online Database, or GOLD, from the JGI. And then we leverage ENVO, or the environmental ontology, for describing environments. So we use this. You can search on the data portal based on the environmental system that you are interested in, following that ENVO hierarchy for ENVO ontology. Okay. So fire hose of metadata importance, but now we're going to do the fun part. So NMDC has a Field Notes mobile app, which is a mobile app that you can take out into the field with you to collect standardized sample metadata in the field while you're collecting your samples. And so I'm going to walk through and walk you through how to use that. So if you follow this QR code, it will take you to a website, which will give you a button to click to download the Field Notes app, either via Google Play Store or the Apple Store. Just Apple Store. I don't know. I'm a Android person. But yeah, so if you want to go check out this app and download it, then you can step through a submission with us, and we'll show you how this tool works. And this kind of gets at that. What NMDC is doing to try to improve metadata capture and improve the quality of metadata. So getting at that, chicken and egg situation I talked about, where, I can't use your data because the metadata is bad. Well, I can't give you good metadata because it's hard. Well, let's make it easy. So we're creating various tools to try and lower these metadata capture barriers and provide information and connections across programs, repositories, and journal requirements, so that you don't have to submit your metadata multiple times. So if you submit metadata via NMDC, then we are trusted brokers within NCBI, so we can submit your NCBI metadata for you. You don't have to do it in both places. Okay, so I'm gonna step through the Field Notes app. So if you downloaded the app, you will first be prompted to log in. When you log into the app, you are required to log in with your ORCID. This is so that we can not only track who generates what, but also so that we make sure that what is your metadata stays your metadata, and that it is private and only connected to your ORCID and your profile. And these, it's the submission portal, and it's the Field Notes mobile app for capturing submission information and metadata, but everything's kept private until you're ready to release it, until you say that, okay, I'm ready for my data to be public, and this is the front door to walk into and share your information via the data portal. The Field Notes app has a little guide on the bottom. So, if you're new to field work, or if you just want some general information, some tips and tricks about what might be valuable to take out into the field with you, that's what that guide is. And then there's various settings there. One important thing I want to highlight here is, like Julia said, we're very community user focused. So we rolled this out with a lot of testing and beta testing to our community and our ambassadors and our champions to get feedback on some important things. So if you really hate light mode, you can change it to dark mode. If you leave your phone open, the app will not close out, your phone will stay on. So feedback like that, just to make the tool more usable and helpful. Okay, so when you first sign in, there is a button to click create a new study. And when you're creating that study, the only information that is required is the study name and the PI's email. So two things that are pretty quick and easy to hopefully capture, just so that we have something on our end to know what that is, but also so that you can find it and come back to it. There is a lot of additional information that you can capture and should capture about your study, obviously, but this is just what's important to be able to move on to the next step. At the bottom there, the next step, after you've added your study name and PI email and hit save, you will be prompted to select a metadata template. These templates, again, are built off of the Genomic Standards Consortium's MixS metadata template. So if it exists in MixS, with the exception of human, you can select that template within our app and our submission tools, and you can select as many templates as you need. So if you're going out into the field and you are collecting, bulk soil as well as plant leaves, you can do all of that in your one submission. So Slido question, what sample environments might you choose, or what type of samples would be important to you. Has anybody not familiar with, or raise your hand, I guess, if you are familiar with GSC and MXS standards? MXS, yes, yes? If you are, raise your hand if you are. Yeah, yeah, okay. Awesome, okay. Water, yep. Yeah, I might almost be able to list most of them. So they provide metadata templates for soil, sediment, water, plant-associated, miscellaneous, natural or artificial environment, air. There's a bunch of human ones that I don't know. There's a lot of agriculture ones, animal ones, host-associated. So there's a wide variety for capturing any kind of microbiome. Soil and water. Okay, I'm gonna keep going. Okay, so once you've selected your metadata template and you are ready to save that, you can add you can select your metadata field. So again, some of the feedback that we got when piloting this was, again, if you've looked at, for those of you who have looked at MXS before, there are a lot of metadata terms. There's a lot of metadata fields. It's kind of overwhelming, and most of those are not relevant when you're in the field. And so here you can select which fields you actually care about and which ones are going to be relevant for your sampling campaign. So you select your metadata fields, hit save, and once you have saved those common fields, you can begin to add a sample. And when you collect that sample, you again will only see the metadata fields that are important, but the thing that we really want to highlight here is that we have incorporated some auto capture tooling. So if you are using the Field Notes app, you can say, use my GPS location on the app, and it will automatically populate your latitude and longitude, and you don't have to capture that yourself. You don't have to go back to the lab and type in your field site into Google Maps and copy paste and worry about forgetting a negative sign, which has happened to us. And so we also have this feature of auto capturing metadata for collection date. So those two things either use, just general cell phone information, and importantly, this can be used offline. So you don't have to be connected to the internet. As long as you've started your submission, you can use the Field Notes app offline. Okay, so with that in mind, what other metadata fields would be of value or provide the greatest benefit to be auto-populated? If you were, if you're either collecting samples or if you are trying to reuse somebody's data and they've collected samples, what do you think would have been valuable for them to have been able to auto capture? Support roles. Coordinate uncertainty. Coordinate uncertainty. I hope there isn't uncertainty if you use the app, because it should get exact coordinates for where you're standing. But if you didn't use the app, that would still be definitely of value to have within like a later collected data set. What is support roles? Anybody want to mention what was that?
Yeah, so I was thinking, like the data creator, data collector, anyone who has access to that data can automatically be added into the data set.
Yep.
Maybe it wouldn't be clear what role they should have, but it could automatically add them in as a point of contact, and then they can go in and make that change to anything other than that.
See that on the next tool. That makes me feel good. Date and time. Yeah, time is a big one that we have been thinking about a lot, because how do you manage time across time zones? And in what format? We've always asked, do we need to normalize to UTC, or do we just provide location? Do you assume it's noon wherever you collected? Yeah, does anybody have any thoughts about what is most helpful when you're thinking about data reuse? Would you care more about everybody using the same time format? Julia?
So I would think that the important thing for reuse has more to do with the daily cycles, so like diel cycles of these microbial communities. And so it's not the time so much as like the relative time of day. So
Yeah.
it's sort of like, yeah, you want to be able to normalize to be very specific about what time zone you're in. But you also like, it's almost like that's not the critical piece. Like I need to know if this is.
If it's at noon or 5 in the morning.
Is it June and noontime? Or are we doing this in the middle of the night? Are we doing this right after dawn? That sort of thing.
Yeah, that's valuable feedback, because, that's what we've been trying to ask ourselves. Okay, I'm gonna go on. So that was a quick walkthrough of the Field Notes app. So again, an app that you can collect your standardized and structured metadata on your phone during sampling. And now I'm going to show you the NMDC submission portal. So the Field Notes app and the submission portal use the same backend. They also follow the same schema structure and schema standards. So everything that you do on one aligns with the other, and everything that you see on one, you can see on the other. And so this NMDC submission portal is available at data.microbiomeData.org. So that will take you to our data portal, like Julia showed. And then at the top is a products button where you can drop down and click on the submission portal, and it will take you to that specific tool. Also, still have to log in with your ORCID, because again, that's how we keep everything connected. And when you first sign in, we have a lot of documentation and videos that you can watch that will walk you through how to create a submission and how to share data. So if you didn't make anything or start anything on the Field Notes app, you can create a test submission that you can start in the submission portal. But if you did use the app, the submission that you started should be visible on the submission portal itself. So again, you can see that, that submission in both places. So the first thing that you see on the submission portal is study information, the same information that you saw on the Field Notes app, to make sure that we have a study name and who the PI for that study is. But there's also description fields, and there's other information about your research there. And it's all important, right? Description, that is what I'm pulling out here to just say that we're really just looking for like a short abstract, something to quick summarize your study, your research, and what you did, and why people should care, or what your target was. Also on the study page, we provide a place to credit your funding sources, contributors, so kind of what you talked a bit about who are the people who contributed to this study. We use the credit ontology to assign contribution level. So did this person participate in sample collection, statistical analysis, data generation? And then if you provide their ORCID, you can also give them permission to this study. So, sometimes the person who has the study information is not the same person that has the sample metadata. So you can all contribute at different levels. And then external identifiers. So again, because NMDC is not a data repository, we're kind of a data discovery tool. We provide links out to existing data. So if you have data in Pride or in SRA or wherever, you can give us those identifiers and those links, and we'll share those so that people can find your raw data, or they can make use of the process data on NMDC. I'm gonna skip that and just ask, is anybody here familiar with DOE user facilities? Just wondering if you've heard of them before or know what they are. No, not at all, okay. So with the no, not at all, I'll just start while we're getting polls in. So DOE user facility is basically a DOE funded facility where you can write in a proposal, and if your proposal gets accepted, you get to use our fancy instruments to generate data for free. So we provide that service to the research community to provide access to these high resolution mass spec data, or really complicated data generation from expensive instruments, as well as expertise for analyzing that data. So that's what DOE user facilities are. And NMDC is close collaborator with EMSL and the JGI, Joint Genome Institute. And so when you submit metadata when you're submitting samples to those user facilities, you have to complete standardized metadata. And it's very different depending on which user facility you're going to. And so NMDC collaborates really closely to provide a resource to capture that metadata, again, in one place. And as I said, we are a trusted broker for NCBI, and we hope to expand this utility across other data repositories so that when you're trying to capture this metadata, you don't have to do it in multiple places. You don't have to learn multiple standards or structures. You can just submit to NMDC, and we can facilitate that sharing with you, or sharing for you. Okay, so here is a list of the sample environments that you could have selected from the app. I think I did a pretty good job remembering most of those. And so when you'll select your sample environment, and then the last step, you will be on the NMDC submission portal tool. So here it looks kind of like a spreadsheet, because that was also from feedback that we received, that having this structure is what researchers are comfortable with. They know how to use Excel. They're comfortable with a spreadsheet. They don't want to fill out JSON. They want to just use a spreadsheet. And so we've kept that structure, and this tool provides in-sheet validation. So when you fill out, if you fill out collection date as June 25th, it's going to say, no, it has to be 4 digits for the year, 2 digits for the month, 2 digits for the date. And you get that immediate feedback that your metadata is invalid and needs to be corrected. And we find that activation energy of keeping people, you know immediately that it's incorrect. You don't have to upload it, wait for an email, be told that your metadata is not valid, and then start again is really important for capturing standardized metadata. I'm gonna just show you all of these boxes at the same time. There we go. So in the NMDC submission portal, we have that's this little bar on the right that pops out that provides some tools to make metadata easier. So if you're looking at a spreadsheet and it asks you for some random string of letters that maybe means something to someone, whoever made that standard to start with, may not make sense to you. And so the column information tool tells you upfront and immediately, this is what this is looking for. Here's an example. And maybe a regular expression isn't the most helpful to humans, but sometimes it is, of what that format should look like. We have a metadata suggester tool. So similar to the app of get my geographic location get my latitude and longitude based on my phone's location. If you then come into the NMDC submission portal, we will suggest an elevation based on the latitude and longitude that you provided. And so this is the first of many aspects within this tool that we hope to expand, of being able to suggest metadata based on other metadata fields or information. We have a find and replace tool that works just like Excel. If you don't want to use the UI, you can download the metadata template and upload it and fill out your metadata via Excel. And then last, we are here to help. We are here to support. So if you find a bug, if you find something confusing, if you don't know what you're doing, you can use these quick access tools to send us feedback and reach out to us. All right, I'm gonna skip here and just say that we were gonna do fill out some metadata, see what you can find, but we're running pretty close on time. So again, just providing the ability for people to validate their metadata quickly is what we are really working towards, to try and lower the barriers of metadata capture, to really get at this connection between what is needed for reuse, but what can be captured. Okay, so I'm gonna open it up again also for some discussion. I know it's getting pretty close. Everybody's kind of winding down. So yeah, just curious, what might be some usability within your community? What would capturing your metadata look like for your community?
So my question is about that upload interface. What size, how many rows do you find that your users upload at any given time? Are you doing hundreds of rows, thousands, 10,000s? What are you typically getting?
Upper hundreds, I would say.
Upper hundreds?
Yeah. We've not yet had anybody be like, oh, my Excel file is too big and it crashed. We did have somebody who uploaded a few hundred samples. at once. So, yeah. If you want to submit thousands and push it to the limit, that would be great. And this is, there's this like column visibility tool because like I said, there's actually a lot, this looks like it's small. There's a lot of metadata fields here. And so we want to provide the ability to be able to focus also as you're filling out your metadata. So, what is required may not be all that's important. And so we try to kind of find that balance between what you have to fill out and what you should fill out. And also, again, trying to provide guidance and help with the community to provide education.
Just a quick question. Could you remind me what standard this is based on?
Yeah, so this is the Genomic Standards Consortium's MIXS, or Minimal Information About Any X Sequence. We collaborate really closely with the GSC. So another important piece that I'll mention here is that if you are using this tool and you find something confusing or hard or unclear or doesn't make sense, or if there's a metadata field that should be available that isn't, we take that information and we take it back to MIXS. So they learn, and then MIXS also is what feeds into NCBI. So NCBI provides the MIXS standard for metadata submission. So we've collaborated really closely with them to transition MIXS to LinkML, and then also figure out how we can improve this for the community.
Could you sort of review what the publication outcome of this is and how people would maybe, so if people are putting a bunch of their samples into NMDC, do they receive a DOI that they would cite in a paper as well? Is that kind of how this ends up working?
Yeah, so we mint persistent identifiers and persistent and resolvable identifiers. So if we've had people share their metadata via NMDC and share that as a source for the metadata on their publication, because again, we're not a repository, so you do still have to submit to NCBI or PRIDE or Massive. Yes, but you would cite them both for the raw data and then the metadata or the process data. And we can submit that for you so that you don't have to deal with an NCBI submission.
Yeah, I guess maybe I just missed that. So where's the breakdown or separation between the metadata and the data? I guess, because as soon as I saw a table, I was like, oh, that's data.
Metadata is data.
Yeah. So is that a separate
Yes,
resource?
so, yeah, so this is the starting point of capturing metadata, again, because we want to provide what you actually truly can reuse, right? So we will not just take any old study with no metadata and dump it into our data portal, right? You have to do this step first. And so, yeah, so with this step, once we get the valid metadata and we are confident that, we have everything that would be needed for truly fair data and shareable and reusable, then it's up to the submitter to decide when their data is ready to be public and ready to be released. If your data already exists on NCBI, we just need that identifier. We'll pull in the data. We'll process it through our workflows. Data and metadata can be shared at the same time or separately. And then if you don't yet have your data published, we just need to know where we can get it, and then we can push the information to NCBI for you.
Do you keep track of metadata at all? So like any fields that you try to assistively auto-populate? Or like when somebody is filling out this portion of the template, is there a place to leave commentary that's like, I actually wanted this thing, but for validation reasons, I chose something else? Do you keep track of any of that?
Yeah, so there is a catch-all column that's just open text where you can provide that kind of, just a string of information if that's what you need to do, because it's a learning process for all, like my background, soil microbial ecology. The soil template is quite intuitive, I think. If you look at the air template or the water template, it could use some work, right? So like maybe getting some of that feedback and like also having that open-ended column of information where if I was to read that and be like, oh, we actually have a column for that, it's maybe just not obvious to the submitter. That's knowledge that we take back to Mixes to make sure that we improve the standard. And then your first question about like metadata, right? Did you use the suggester tool to fill out your elevation, or did you know that yourself? We are going to be thinking about that as we think about more metadata suggester tools and applicable fields, and leveraging AI to, again, like, here's my publication. Can you fill out some of that metadata for me, right? If it's obvious in my publication that I had 20 samples, 2 temperature treatments, can AI fill that out? And if you exclusively use AI, what is the rating that you get versus like if you fill it out yourself and it's totally manually curated, right? What is that balance? So that's something we are going to start thinking about.
This is really cool.
Thanks.
My question's kind of related to Jim's, and it's, I think you said you can share your data, you can share your metadata. It doesn't have to be at the same time. Is this a requirement now that it exists for people to submit this when they, not at the exact same time, but to have their data submitted?
So it is a requirement for the DOE user facilities for the way that they are capturing metadata, and then it's a requirement for NMDC. If you want to go to NCBI and fill out the Excel template without all the helpful tools, you can still do that. You don't have to use our tool, but if you do, you get great metadata, and we will submit to NCBI for you. So no requirement based on like the repository level. This is just us trying to lower the barrier and provide tools to make it better. Thank you.
And the validation too is if you put in something that's incorrect and you hit that validate button, it highlights it in bright red and says this is not valid, or so if it's invalid or if it's missing, that always is my favorite feature of it. Makes it really obvious when you're validating what you need to improve upon to get it submission ready.
Okay, I had a couple more Slidos, but I think I'm gonna just skip through them. And I'll just say, Julie, do you wanna give a quick wrap up?
Yeah, so thank you all for joining us. Just to wrap up, we wanted to talk about the quick jump between metadata and comprehensive metadata and AI readiness, and how we kind of think that this will go hand in hand as we move forward. So we thought providing you with some of the resources and tools that we've developed or that we think are important to all of you to also be thinking about how they could apply to your data types, and then what that means of if you have perfect metadata, is that more AI ready? If you have half of the metadata fields, is that still AI ready? So just kind of thinking about for your own data what that really means, and then this whole you are what you eat of if you're comfortable with bad metadata and bad AI insights, that's okay too. But just kind of thinking about this garbage in, garbage out process. And again, our data types are sequencing data and mass spec data. So metagenomics, metatranscriptomics, metaproteomics, and metabolomics. And then our metadata is kind of what all these other fields are. But I liked the comment of one scientist's data is another scientist's metadata. So having those connections here and what that really means for making sure that everything is metadata rich. And then, yeah, we also just, if you get nothing out of this session, maybe that microbes are really cool and maybe you should think about them in your work. And then, yeah, we know that you all are doing really cool work. So then just also thinking about how things like reporting standards for your manuscripts might fit into your current workflow and things that might save you time rather than just kind of adding on to your time. And then, as we said, we're super community driven. So if you have feedback about the app or the submission portal or the reporting guidelines or anything else, let us know. The app was kind of born from what we thought were the needs of the community members who had pieces of paper fly off of a ship that had their metadata on it, or someone leaving a notebook in the desert that also had their metadata, things like that. So trying to help the community, but we have to do that in a way that is meaningful and works for all of you. So if you have feedback also let us know. I'm not going to take this candy home, so if you all want some more, please come grab some. And thank you all for joining us on a late afternoon Thursday session. But thank you so much.